In this video we will discuss about this adipose manipulation transplantation can suppress cancers in the in vivo studies also. So how they did the in vivo studies and what are the results, we will discuss that area in this video. So they took this uh, breast cancer patient samples, and in this they took out the nearby breast adipose tissue and the breast tumors, and then what they did is they prepared the organoid of these breast tumors and co-culture them with this adipose transfected or which are transduced with this UCP1 gene, which can uh, so this adipose can overexpress UCP1 genes that will cause them more nutrient consumable adipose. So when they co-culture them and then they check this organoid size, organoid number, proliferation markers and tumor metabolite profiles, and what they are able to see in terms of this organoid size is that organoid size goes down when these tumors are co-cultured with this adipose, which are transduced with UCP1 genes, compared to the control where they are transduced with VP64 gene. So, and in terms of the number of organoids also, in most of the cases the number of organoids goes down except for this TOR 124 and TOR 41, and they did their studies on 5 different breast tumors and adipose tissues. And when they check the proliferation markers in terms of this MKI67, it also goes down significantly in all the 4 cases when they check the relative mRNAs levels. And thereafter they check the relative mRNAs levels of this uh, glycolytic pathways, that is GLUT4 and GCK genes, and the fatty acid oxidation pathway, that is CD36 and CPT1B. In the previous video I might have mislabeled them as proliferation markers, but they are not proliferation markers. This GLUT4 and GCK belongs to the glycolytic pathways, and CD36 and CPT1B belongs to the fatty acid pathways. And in all these 4 cases they are able to see a significant decrease when they co-culture these tumor cells with the adipose, which are transduced with UCP1 genes. Not in 100% of the cases, but in most of the cases. For example, in TOR 41 they do not see a significant change, and similarly for this GLUT4 and CD36 and CPT1B, they do not see significant changes for this TOR 41, but in majority of the cases they see changes, and the relative mRNA levels are lower in the case where tumor cells are co-cultured with adipose, which are transduced with UCP1 genes. And thereafter they wanted to check this is the in vitro results of the patient samples. Now they will check the in vivo results, that is in the mice models of this patient samples data. So they did the same things, that is they took out the breast tumors and prepare organoid and implant them into the mice model, and after 3 weeks they implant these breast adipose tissues, which are uh, modulated in a way that UCP1 genes is overexpressed. So after 3 weeks they implant them, and then after implanting of this adipose, they allow the mouse to grow for 3 weeks, and then they check the tumor volume, growth, metabolic genes, hypoxia, angiogenesis, and in terms of this tumor weight they are able to see a significant decrease when the tumors are co-cultured with this adipose with UCP1 gene overexpressions, and the proliferation markers also goes down in the compared to the control case, that is MKI67, and the fatty acid fatty acid related genes such as CD36 and CPT1B, and glycolytic related genes such as GLUT4 and GCK also goes down significantly. Then and this is the bottom side of the panel F is the quantifications of the tumor weight. You can see that it is also significantly lower when the tumors is co-cultured with this UCP1 upregulated adipose. Now they wanted to check whether their strategies also works for the high risk cancer cells or not. So they take the samples from this BCRA1, 2 and RAD51 mutated carriers and did the similar studies, that is in the in vitro form, that is make an organoid and co-culture them with the adipose, which is also from the same patients and in which UCP1 gene is overexpressed, and thereafter they check the organoid size, number, proliferation markers and basal or luminance markers. So in terms of the organoid size, the results are similar, that is when the tumors are co-cultured with the UCP1 gene overexpressed adipose, they see a organoid size is lower compared to the control where it is co-cultured with adipose in which VP64 gene is overexpressed, which does not have much impact on the nutrients requirement. And in terms of the organoid numbers, they are able to see significant changes for 2 patients out of the 3 patients. And then when it comes to this proliferation marker, and they are able to see that MKI67 is statistically lower in the case where tumors are co-cultured with adipose with UCP1 genes overexpressed. And they also check the other markers such as mTOR, CK5 and CK17. All of them are lower for all the 3 patient samples that they have used. So all these 3 cases indicates that all the similar results from the 3 cases indicates that their adipose manipulation transplantations has a therapeutic impact in terms of the suppression of the cancers, and that they have showed using the in vivo studies from the patient samples and also the in vitro studies from the patient samples, and they even shows their results are consistent even for the high risk breast cancer in vitro studies. So this is how they validate their hypothesis about adipose manipulation transplantations. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.